Hello, this is Raymond Lam from the Department of Biomedical Engineering in the City University of Hong Kong. I'd like to introduce some works from our research team on the automated microfluidics for bioapplications. Microfluidics is the collective technology miniaturizing devices handling liquid in a very small scale. The direct benefits from it is on the much shorter time of transfer of mass and heat. The microfluidic devices generally would have the advantages on speed, precision, and less consumption comparing to the bulk macro-scale devices. Microfluidics can also provide some extra functions to the device that the macro-scale devices cannot offer. Because of the small length scale comparing to different parts of the human body, microfluidics can mimic some environment in the human body. In fact, many cells in human body are experiencing fluidic microenvironment. For example, the blood circulation systems, including the heart and lungs. One direct application of microfluidics is to replace the laboratory works, so that we do not need to use the pipetting. Nowadays, there are already quite a number of commercial microfluidic systems in the market. And some of these commercial products can replace the existing biomedical instruments, for example, the PCR thermocycler and the flow cytometer. On the other hand, microfluidics can replace the manual efforts by automating the entire microfluidic system. The right photo shows an example of a microfluidic device automating the pipetting process that we need to work with the well plates. The device shown here is filled with different color dyes for better visualization. The area of this device is about 3 to 4 centimeters times 2 centimeters. We can see it contains 32 microchambers in the middle, replacing the 32 well plates. And we can also see there are some microchannels around, and in fact, there are multiple layer channels. Back in the old days, I learned automated microfluidics under the supervision of Dr. Todd Thorson while I was studying for my PhD in the Massachusetts Institute of Technology. The basic control elements of microfluidics are microfabs, micropumps, and the multiplexers. They are controlled by air pressures. The air pressures can be supplied and controlled by an external system, and the device can be placed and being observed directly on the microscope. The basic working principle of the microfab, as shown at the lower left corner, is about 2 layers of structure. The upper layer is flowed with the air, and it has a dead end, and therefore if we connect that to the air pressure controlling system, this part can be pressurized, while the lower layer is flowing with liquid, is the microchannel. The microfluidic device is typically fabricated with silicone material, which is biocompatible and flexible, and therefore if the upper part is pressurized, the silicone membrane between the 2 layers would be formed and in fact being pressed downward, closing the lower flow channel. And by pressurizing and unpressurizing the upper layer, we would be able to open and close the lower flow channel. So this is the working principle of the microfab. By arranging multiple microfabs in different ways, we can achieve the peristaltic pumping as well as the gating as the multiplexing manner. Let's take a look at some examples that microfluidic device can achieve. In this video showing on the right, it shows the process of inserting a liquid in blue into those 32 chambers one by one. and afterward the microchannel around will be washed. So it's like the pipetting process of a liquid onto a 32 well plate one by one. And the entire process is automated. And in fact, we can control the liquid insertion in only one particular microchamber. The video at the lower left corner is showing the mixing process achieved by multiple microfabs. So we can apply it in each of the chamber in the 32 chamber in the microfluidic device. With such microfluidic device architecture, we can scale up the device to have more chambers, for instance, over 100 chamber. And we can see that this 128 chamber device can be filled with different colors at different locations specifically. So it's showing the word teeth here. In fact, there are many factors that can be controlled by the microfluidic device. So their liquid compositions, substrate material, substrate stiffness, substrate coating, dissolved gases, mechanical stretch, flow shear stress, cell deformation, cell-cell interaction, cell structure interactions, and etc. At the lower right corner, it shows the cell growth under different dissolved oxygen concentrations. There are many applications of the microfluidic device. Some of the applications would be on cell sorting, drug screening, or any cell-based assays. Furthermore, we can design more specific microstructures in the microfluidic devices to achieve a specific task. For instance, we have developed another device that for characterizing the biomechanical properties of floating cells. The direct applications of such device would be for testing cells in the bioliquids. So circulating tumor cells or other cells in blood would be our target testing cells. The idea was originated during my postdoctoral training under the supervision of Professor Jianping Fu in the University of Michigan. The device layout is shown at the lower left corner. It contains a basic element, which is the confining microchannel. So the working principle is showing as the picture on the right. So we flow the cells along this confining microchannel, and because of the channel width reducing along the flowing direction, the cell would then be clamped and compressed. And based on the cell deformation, we can figure out the biomechanical cell properties. At the same time, we can functionalize the channel walls with a particular antibody so that the attachment or adhesion of the antibody with the cell would be able to reflect a certain surface protein expression of the cell. The video at the lower left corner is showing a cell passing through a confining microchannel. This is the other microfluidic device we developed for cell sorting. The early stage of this work is in collaboration with Professor Marcos from the Nanyang Technological University in Singapore. It applies multiple microsieves arranged in a specific way to achieve the sequential cell trapping and sorting. Once again, we can functionalize the structure walls with a certain antibody to capture cells with a certain surface protein expression. One direct application of such device is for isolating circulating tumor cell in blood of cancer patients. We can take a look at the video playing at the lower left corner. We can see that those particles can be trapped one by one sequentially along the flow stream. So in these couple minutes, we have gone through a few microfluidic devices developed in our research team, and we hope you enjoy it. Thank you very much.